So thank you. It gives me great pleasure now to turn to the main event and introduce our distinguished lecturer for 2026, Professor Marlene Behrmann. The idea of honoring Marlene received incredible support from our members, and I was really very, very glad that we were able to get her to accept our invitation. She is truly an innovative and successful visual neuroscientist, and I think we're in for a treat. Marlene has a very long CV with lots of records. I'm going to try and highlight some of these. Marlene holds the John and Clea Shepard Chair in the Department of Ophthalmology at the University of Pittsburgh. Her work studies the physiological and neural basis of visual processing with specific attention to the mechanisms which the signals from the eye are transformed into meaningful percepts from the brain. She adopts an interdisciplinary approach combining computational, neuropsychological, and neural imaging studies with adults and children in both health and disease. For example, she has mapped the cortical system in patients following childhood hemispherectomy, which she'll talk to us about today. She has received numerous awards. She's a member of the Society for Experimental Psychologists, the National Academy of Science, the American Academy of Arts and Science, she's received the APA Distinguished Scientific Career for Early Career Contributions, and the Kavli Distinguished Career Contribution in Cognitive Neuroscience Award from the Cognitive Neuroscience Society. Please join me in welcoming and inviting Marlene to give her lecture.
I want to be sure that you can hear me. The microphone's working. Yeah, okay, great, thanks. Let's be sure that. I'm grateful for the opportunity to participate in this memorial symposium honoring Professor Ian Howard, and would like to thank the organizers very much for the invitation. Thank you, Rob, thank you, Laurie, thank you, everyone. I stand here today on the shoulders of giants, not only those of Professor Howard, but also the shoulders of the very many people at York who have made such meaningful contributions to the field. I also want to recognize Dr. Morris Moscovich, my PhD advisor, who is here, and without whom I would not be here. I had the opportunity to learn more about Professor Howard's contribution to science while preparing for this talk, and his sojourn in Toronto, contribution to the development of the Department of Psychology, and the founding of CVR are especially important and notable. In reading his obituary, I noted that the family requested that donations in his memory be made to Doctors Without Borders, an organization that I'm really happy to support. And I'm pleased to have had an opportunity to contribute in his memory in recognition of today's memorial event. But I also learned, as Rob has already alluded to, that it's not all work and no play for Professor Howard. Here's a photograph of him dancing with his wife, Toni. Everyone speaks of his kindness, his fun, and his curiosity. And everyone makes reference to these annual events that include parties with puzzles and toys. There's also some kind of zip line that deposited the rider in the trees, and some other apparatus in which participants sat not knowing that they were unknowingly in an experiment where somebody ran round and round until they were almost horizontal and spinning, and they really thought that they were stationary and it was the visual world that was really changing. So really sort of creative, innovative, and exciting ideas for a lab party, all of which I'm going to try and adopt for my own next party. So I was here at CVR in the summer of 2025, so not a long time ago. And I'm really happy to be here again. They did give out these kind of t-shirts from last year in the summer. It says the brain and integrative vision, and it's a great t-shirt. I was thinking about wearing it today, and it's like, well, it's not quite summer in Toronto yet. Okay, I also decided, given that I had spoken here in the not too distant future, that I would talk about a completely different topic. And today's overarching topic will address the organizational principles that underlie brain behavior correspondences. So on the one hand, that's in general and in the visual system in particular. So on the one hand, there are theories such as this one by Gall, in which the argument is that behavior emerges from specialized, segregated regions, and here you can see Gall doing his experiments. An alternative view espoused by scientists such as Lashley have challenged this one-to-one correspondence and favored a view in which behavior is mediated by more distributed and widespread cortical circuits. So this debate has raged for hundreds of years now and is humorously referred to as the skull palpators versus the bird brain ablators. And the essence really is whether we should be thinking about behavior as mediated by a set of segregated, specialized regions or an emergent property of distributed, integrated circuits. Somewhat more contemporary versions of this debate come from Brodmann's anatomical cytoarchitectonic maps of these segregated regions, through Foda's thesis of modularity of mind, but also more recent structural and functional parcellations based on multimodal imaging data. In many of these accounts, there's also the built-in assumption that these divisions are innately pre-specified and not really amenable to change. The opposing, more contemporary perspective reveals that at different scales, even local scales, you can see distributed circuits such as orientation pinwheels. We can also see gradients across retinotopy, but also widespread whole brain gradients during resting state and tiling of the brain across these semantic maps. These ideas clearly indicate that brain organization may not be so neatly divisible. So in today's talk, I'm going to examine the evidence for these opposing perspectives using two well-known and well-loved examples of segregation in the cortical visual system. So the first one is very obvious, and that is the segregation or the functional division of labor between the visual systems of the two hemispheres. As you undoubtedly know, the long-standing view of the hemispheres, captured well in this media image, is that the right hemisphere is creative and holistic, as depicted here by all these colored flowers and their kites being flown, whereas the left hemisphere, in its analytic fashion, has these faceless people working in small monochrome cubicles. The second distinction concerns segregation of function within a single hemisphere. And this specifically refers to the claim of the two visual pathways, one dorsal and one ventral, each of which have different functions. And this is perhaps well depicted in this now quite old but very nice visualization, in which the dorsal visual cortex that projects up to parietal cortex is responsible for representing spatial positions in the visual input, whereas the ventral temporal pathway is specifically responsible for coding the geometry or the shape of the information. And sort of never the twain shall meet in a picture like this. So I'm going to take each of these examples in turn, the between hemisphere and then the within hemisphere. I'm going to start off by showing you the evidence that supports segregation of function, not just pretty pictures from the media. And then I will switch to turning to the distribution of function, and then I'll be able to wrap up and reach some conclusions. So if we think about the between hemisphere division of labor, this is a somewhat more realistic depiction of the way that we might think of the division today. So word recognition as a property of the left hemisphere and face recognition as a property of the right hemisphere. And the empirical support for this is really quite strong. So for example, in neuropsychological studies, a lesion to the right ventral area will result in a deficit for faces known as prosopagnosia, and a lesion to the homotopic region in the left hemisphere will give rise to a reading deficit, a deficit for word recognition, in previously literate individuals. But it's also the case that electrophysiological data, for example, mapping the pattern N170 waveform, shows that there is a greater N170 over the right hemisphere for faces and over the left hemisphere for words. And lastly, over the last few decades, with the advent of functional imaging, we've seen the demarcation of circumscribed regions, for example, here in the right hemisphere for face recognition and one in the left hemisphere for word recognition. So the evidence is strong and weighty, and in fact is well supported by many more recent studies which place participants in the bore of a magnet and then shows stimuli like words, faces, objects, bodies, houses, etc. onto a screen in the bore of the magnet. And then the magnitude and the site of selectivity of the neural response for these different categories is quantified. This is a standard result that we obtain, everybody else obtains the same result. First, let me orient you. This is the inferior surface of the brain. So if I took a wire cutter and cut my head off and flipped it over, you'd be looking down on the base of the brain. I've stripped away the meninges, the callosum, the cerebellum, etc. And this is just the inferior inflated surface. And the schematic is chosen specifically so that we can see all of the activation from both the gyri and the sulci shown in the same plane. And the important result here is that there are different regions shown here in these different colors that show selective responses to these different categories. And I'll draw your attention to, again, the right hemisphere activation for faces and the left hemisphere activation for words. So here we have weighty evidence supporting the segregation of face and word representations to the two hemispheres. Close inspection of the imaging data, however, show that there is, in fact, these are 4 different studies. This actually is a schematic, I should have used the actual study, showing that there actually is bilateral activation. So even in the original sort of seminal study that identified the fusiform face area in the right hemisphere and has a rectangle around it, there actually is also activation in the left hemisphere. But if you subtract the left activation from the right activation, you're only left with the right activation. But really, the activation is bilateral. And that's true in all of these cases. The same is also true for words. So this is the sort of paradigmatic PET study that identified the visual word form area with its rectangle. And of course, you can see in the right hemisphere, there is also activation, just not as strong. And we see bilateral activation in all of these regions with a difference in the weighting. And so one of the problems in reaching conclusions about the relationship and the functional division between words and faces is that these studies only ever show the participants' faces or only ever show the participants' words. So a number of years ago, Uri Hasson, Rafi Malach, and I, and a couple of others, decided that we were going to show both words and faces to the same participants. There are 11 individuals here. In the center, again, is the inferior inflated surface. But to show you really the full extent of activation in the brain, on either side here is the flat map. Activation that's for words is shown in purple and for faces in orange. And you will note here that in both hemispheres there is both purple and orange, reflecting the bilateral activation. But here in the right hemisphere, there is more activation for faces than there is for words. And in the left hemisphere, there's more activation in purple for words than there is for faces. Suggesting not only is there bilateral activation, but it has this weighted asymmetry. You might be wondering whether, in fact, the word activation in the right hemisphere and the face activation in the left hemisphere play a functional role, or it's just epiphenomenal transmission of the signal to the other hemisphere. So one way in which you can evaluate this is to, well, the prediction is that individuals with a unilateral lesion to either the right or the left hemisphere should be impaired at both stimulus types, but to a greater extent for one than the other, depending on which hemisphere is affected. So a number of years ago, David Plout and I recruited 3 of these pure alexics with left hemisphere lesions, 3 prosopagnosics with right hemisphere lesions, and we're going to show them, we're going to test them both on word recognition and on face recognition. So in this very simple experiment, they had to simply read words that were either 3, 5, or 7 letters in length. Here are the control data. As you can see, there's no change in response time on the y-axis as a function of word length in the controls. But in these patients with a lesion, we see this monotonic relationship between reaction time and word length, reflecting the signature letter by letter reading, as it's commonly referred to. Having established the basic signature, we can now see what happens to the prosopagnosic individuals. So they've got their own controls, because we matched very carefully to each of the groups, who also are not showing any difference. But here we actually do see also a letter-by-letter effect, a monotonic increase in the prosopagnosics, just not as steep, nor do they take as long in absolute reaction time. But nevertheless, they have an impairment relative to the controls. For the word recognition experiment, I'm going to show you error here on the y-axis. The experiment is 2 faces appear on the screen. They could be the same or different. When they differ, they could be 2 very different faces, or we morph them to some extent, or we morph them to a large extent. So this is an easy discrimination. This is somewhat more difficult because the faces share many more properties, and here in the difficult condition, they share a lot of properties. I'm actually only going to show you data from the easy and medium condition, because in the difficult condition, nobody could actually do this study. This is experimenter over enthusiasm, I think, that had to be sort of reined in. So here we have the data from the prosopagnosics controls. They actually do make more errors, but there's much more variability in the medium than easy discrimination. And the prosopagnosics, of course, show an increase in the error rate as the discrimination becomes more difficult. Here are the alexic controls. They're also showing some difference. And here in the alexic individuals, we are also seeing a difference, almost of the same magnitude as the prosopagnosics, although the overall error rate is lower. So in both of these cases, with focal lesions in adulthood, we are seeing an impairment not only in the major domain, but also in the less preferred domains. So severe face recognition deficits after the right hemisphere lesion, and a mild word recognition deficit, and the complement following a left hemisphere lesion. So here we only tested 6 individuals, and here we only tested 11 individuals. But there does come a time when one has to think, well, to what extent are these small groups truly representative of the population at large? And that's what kept me awake at night, and so I decided, in we go, we're going to scan 55 individuals, which is what we did, as it's not exactly representative of the population at large, they're all college students. But it was a wide range of students, and we scanned them on a wide range of categories. We also had lots of control conditions, inverted faces, inverted words, scrambled, etc., etc. And here is the group map, the average, shown only here on the flat maps of the left and the right hemisphere. Here, for some unknown reason, we decided to put words in blue rather than purple and faces in red rather than orange. You would have thought that it made sense to keep the color palette the same. probably so. But we basically replicate the previous result, and that is there's both red and blue in both hemispheres, consistent with this idea that there's bilateral activation, but it's weighted. More blue than red in the left hemisphere, more red than blue in the right hemisphere. But you might be wondering, why is it weighted this way? I mean, it's all well and good to have bilateral activation, but why isn't it a coin toss as to which hemisphere is responsible for which particular set of representations? And we argued, and then I'll show you some empirical evidence, we hypothesized that brain organization is severely constrained by connectivity. So many of you know the Fellman and Van Essen very famous diagram just of the primate visual system. I mean, it's massive, right? There is so much connectivity here. And all of this essentially has to fit into a skull that's like yay small. So Nelson and Bowers did some back of the envelope calculations, and some of you may know this, I use this example a lot. If you assume that we have 100 million neurons and we place them all on a sphere and we connect them all to each other with a small axonal radius, this actually will result in a brain that is 20 kilometers wide. But in fact, the human brain is only about this big in comparison. So the wiring economy of the brain is really important, and there has to be minimization. So regions that need to cooperate need to be close to each other. You cannot send signals over very long distances. So connectivity must be sparse, and it must be, you want to minimize the arrangement to reduce the spatial costs of the connectivity. And so when we looked back at our data, in addition to looking at the word and face representation, we also noticed that there is this big blue region right here in the front. And this, in fact, is Broca's area. So when we read, we aren't only trying to derive a good percept of the input, or the orthographic input. We're actually trying to understand what it is. And so connectivity and coupling to language areas is really important. And so the claim here is that the pressure for Broca's area and extrastriate visual area to cooperate gives rise to the emergence of the left visual word form area, because language is present in the left hemisphere in the majority of individuals, all the right-handers and the majority of the left-handers as well. So the claim here is that the pressure for visual word areas and language to reciprocally send signals is going to drive the emergence of literacy into the left hemisphere and not into the right hemisphere. So we also did many computational studies that examined connectivity constraints in artificial neural networks, large models, and we tested very many properties. For those of you who have any interest, it's published in 2022. I'm not going to go into a lot of details, but the idea here is that we showed that this wiring minimization, this economy of distance, can lead to the discovery of properties of inferotemporal cortex that is very similar to what we see in humans. But we also went to get empirical evidence. We needed data to support our claim about wiring pressure. And so specifically, I'm not sure why this is off to the side here, specifically the claim that it is the acquisition of literacy that's going to drive the pressure for the coupling. So the hypothesis is that in young children who don't know how to read, they're mostly fine-tuning both hemispheres for face recognition. When they begin to go to school and develop a little into early adolescence, we can see that by virtue of language being in the left hemisphere becomes more tuned for orthography than the right hemisphere, because it needs to couple language areas together with extrastriate areas. Both hemispheres have got both words and faces, but the left hemisphere is now on the path to being tuned to be much more orthographically sensitive. And finally, in adulthood, we have the bilateral weighted asymmetry that I've been talking about, and this is the hypothesis. So the data, some data that we collected here, comes from really the simplest experiment that you can imagine. They are in a chin rest, they're looking at a screen, there's a fixation, a face comes up, a very brief fixation again, only 150 milliseconds to make sure that the eyes do not saccade from fixation, and then a stimulus, second stimulus comes up in the right or the left visual field, and they're making same different judgments. We do this for faces, we do this for words. And here are the behavioral data. So accuracy here is on the y-axis, and this is just for the adults. So shown here is the significantly higher accuracy for words when they are shown in the right visual field, i.e. in the left hemisphere, than in the left visual field to the right hemisphere. And here we see the complement of that, so higher accuracy for faces in the right than in the left hemisphere. And this is the adult bilateral weighted asymmetry that you are getting tired of hearing about. But if we look at the young adults, we see that in fact they don't show this kind of complementary lateralization. They're only showing the lateralization for words greater in the left hemisphere than the right hemisphere, and no difference for faces. And in the youngest children who've just learned to read, even though the overall accuracy, of course, is much lower than the other two groups, they too are showing lateralization for words but not for faces. And this supported for us this idea that it's the earlier emergence of this lateralization that's going to drive the optimization of orthographic representations in the left hemisphere. So the claim here is that it's the pressure to read that drives not only the tuning of words in the left hemisphere, but pushes out faces largely, but not exclusively, to the right hemisphere. We also collected independent data just from these young adolescents and the children on a standing reading comprehension measure, independent of the experiment. So on the x-axis here is how good they are at reading, and on the y-axis is their degree of face lateralization. So the first thing you notice, it doesn't matter what age they are. You can see the two symbols are kind of intermixed. This is not an age effect, but what it is the better you are at reading, the greater the lateralization of faces to the right hemisphere, suggesting that there is this kind of push-pull complementarity that is going on, this interdependence of the two hemispheres. And we collected EEG data, this time just from the adults and from the children in that study. Plotted here is the N170. It's supposed to be negative, but I'm plotting it up so that the graph looks the same as the behavioral data. And here we see, as we expect, lateralization for words to the left hemisphere and for faces to the right hemisphere, and the lateralization for words only in children, but not for faces. So the electrophysiological data support the behavioral data that I've already shown you. So most of what I've told you rests on this idea that it is the acquisition of literacy that's going to drive this coupling by virtue of the wiring minimization principle. And in fact, that was our story for a long time. And unsurprisingly, it was consistent with so many other studies showing these language lateralities, Broca's area and Wernicke's area along here. But in studies like this, including the one by Reza Rajamir and colleagues, they also show lateralization in the right hemisphere, but for social behavior. So not just face recognition, but emotion recognition, biological, especially animate biological motion. And so it compelled us to now think about whether or not there was equivalent pressure coming from the right hemisphere. It wasn't solely a function of the acquisition of literacy. And when we went back and analyzed our data, Here you can see the superior temporal sulcus, sort of the major area involved in tracking eye gaze and eye movements, emotion and expression, and showed that there is a tight correlation between STS activation and face activation, just like there is tight correlation between Broca's activation and the visual word form area. And so it's not solely a result of what's going on in the left hemisphere. There is also right hemisphere pressure for the same minimization principle. Okay, so I've told you that there is distribution of function. Both hemispheres appear to have both word and face representations. They're asymmetrical. There are different pressures in the two hemispheres that gives rise to this emergent mature topography. But before we can make this claim, you might be wondering, or there's an alternative, much easier, much simpler hypothesis, and that is what we might be seeing in the other hemisphere. Let's just do it over here. What we might be seeing in the other hemisphere is simply the propagation of the signals across the corpus callosum to the other side. I mean, the corpus callosum has got 200 million axon wires. And so signals are being pushed around in the brain extremely rapidly and actually with significant faithfulness, veridicality. And so how do we know that whatever face recognition we're measuring in the left hemisphere might simply just be the cascade of the computations in the right hemisphere? So is this all just an artifact of propagation? So I'm going to show you some data that I think rules out that simpler, sort of tantalizing explanation. And I'm going to show you data here from individuals who only have a single hemisphere. So these are individuals who have had extensive resections of the brain, in fact an entire hemisphere, hemispherectomy, for the management of drug-resistant epilepsy, and these are all done during childhood. And these individuals grow up with a single hemisphere. We were fortunate to be able to recruit 24 left hemispherectomy and 15 right hemispherectomy patients to participate in the study. And the very first study that we did was to examine behaviorally, using the same experiment that I've just shown you, what a single hemisphere is capable of, and whether or not that depends on whether they have a preserved left or a preserved right hemisphere. So I've already told you about this very simple and easy kind of experiment. Let me show you the results. Again, accuracy is here on the y-axis. This is just for faces. This blue here are the controls. We had two groups of controls. The lighter dots are the younger controls. The darker dots are the older controls, because patients are both a little bit younger, like 8, and then the others who are like 13 and 14. So we had two separate control groups. So on average, the controls are scoring around 92%, which is pretty good. And here are the data from the patients who just have a left hemisphere or patients who just have a right hemisphere. And you can see that on average, they're scoring about 85% correct. So they are statistically inferior in their accuracy compared to the controls. But it doesn't seem to matter whether the left or the right hemisphere is preserved. And when we looked at the word data, we have the exact same finding, and that is statistically inferior to the controls, but nevertheless reasonably good performance at 85% correct, irrespective of whether they have just a preserved left or preserved right hemisphere. So it turns out having one hemisphere is great if you can get 85%. It's not quite the same as having two hemispheres, which yields closer to 100%, but a single hemisphere has representations for both words and faces to an equivalent degree, independent of the hemisphere. This is both a testament to the plasticity of the brain post-hemispherectomy, but also to the functional contribution of the remaining hemisphere for both words and faces, independent of there being a corpus callosum present. So I think the time has come, having shown you the segregation between the hemispheres and instead the distribution of function from both imaging and even patient data, as well as behavioral neuropsychological data, to now examine whether or not a similar claim about distribution of function might apply even within a single hemisphere. So I'm going to talk to you about the segregation within a single hemisphere into dorsal and ventral functions. Again, I'll just remind you, this is the image that I showed you earlier on. There's been an updated conceptualization where the parietal pathway is not only for spatial representations, but also for action representations. It doesn't really matter for today's talk. I'm really just going to be talking about the spatial representations and the shape representations, so the where and the what. So the original support for this claim came from these studies that were being done by Mort Michigan, Leslie Ungerlein, and some of their colleagues at NIH in macaques. This is the brain of a macaque monkey. So they took monkeys and they trained them, for example, on a shape discrimination task. So in this experiment, two shapes appeared on a tray before the monkey, and underneath one of them, in this case the orange pyramid, a peanut was in a food well. And all the monkey had to do was learn to displace this pyramid and retrieve the peanut, and it made no difference if the pyramid was on the left or the right. The reward was associated with that particular shape. They also trained monkeys to do a spatial location discrimination task, and here the stimuli were the same. They are both these orange pyramids, but only one of them was baited, and the way that the animal learned to know which one was because one of the pyramids was in close spatial proximity to this blue cylinder. So the cylinder could appear on this side or this side, and the peanut was in the food well associated with the closest spatial relationship. And so essentially they then went on to lesion either the temporal cortex or the parietal cortex, and they showed that after this temporal lesion, the monkeys were able to do the spatial, often called landmark task, this one here, but failed miserably on shape discrimination, whereas after a parietal lesion, they were able to do the shape discrimination, but were terrible on this spatial relation landmark task. This is the famous double dissociation that argued for segregation of what and where function within a single hemisphere. There are in fact human data that support this segregation. So here, for example, after a large lesion, a middle cerebral artery infarct to the right hemisphere, patients, not uncommonly, show a disorder called hemispatial neglect, where there is a failure of spatial orientation to the side of space, here the left, contralateral to the lesion. There's something about how they don't quite have the right spatial layout of the input. And after lesion to the what pathway, so to temporal cortex, patients can show visual agnosia, and here, data from one of my patients is a failure to recognize the shape, called this a coconut, this was a cash register, a spider, and didn't know what this is. So this is a visual agnosic patient who is clearly not blind, but somehow doesn't quite get the full geometry and the full shape representation. And basically reflects the what-where discrepancy and segregation in humans. So I just added it into this matrix here. And again, what's really complicated is that people typically don't evaluate both spatial function and shape discrimination within the same individuals. And so this sort of pushes the literature and supports this claim about segregation of function. So a number of years ago, a graduate student, Valentina Zachariou, together with Bobby Klasky and I, decided that we were going to look at both spatial and location information, sorry, both spatial and geometric information in single individuals. So we had two tasks. This was sort of, they had to tell us which of these displays had a shorter distance between the chair and the line. And sometimes there was a longer distance, sometimes the discrimination was more difficult. We also had a sort of parametrically manipulated object-based study. We had to say whether these two are the same or different. These are more different. This is round, this is very spiky, this is round, and this is a little bit round. Okay, so we basically manipulated spatial proximity and geometric properties, geometric overlap. And we saw the following. So this is the lateral surface of the left hemisphere that I'll deal with first, and show you that, so shape here is going to be in yellow and location is going to be plotted in blue. So of course we see very significant and substantial activation for shape along the swath of ventral cortex. And we also see significant and substantial activation in parietal cortex in the location discrimination task. But what was really striking to us was the fact that in dorsal cortex as well, we're getting huge amount of shape selectivity as well, or geometry response in this kind of task. This here is a top-down view. So we're looking at the top of the head, and this is the location information in parietal cortex, and this is the geometric object-based information also in parietal cortex. And that led me to start wondering about the nature of shape sensitivity in parietal cortex and whether or not shape information was distributed not only in ventral cortex but also in dorsal cortex, contra the division of labor within a hemisphere to what and where. So together with Aries Freud, who I know is here and loves being here, and was a fantastic postdoc, and Jody Cullum, David Plout and I, decided that we were going to map shape sensitivity in every visually activated voxel in the brain. And I've really come to grips with the question of how much and where is the shape information. So we ran two experiments. I'm only going to show you the one. In this experiment, we had intact shapes like this, and then we decomposed these shapes into 4, 16, 64, and 256 pieces. And we reasoned that and we're going to measure this in every voxel. We're going to take the beta weight in a single voxel, and we're going to measure how strong the response in that voxel is as a function of the decomposition of the stimulus. So the stronger the beta weight here, and this sort of positive slope from here to here, suggests that this is really shape sensitive, because the better the shape, the stronger the beta weight. So hypothetically, that's one voxel response. Another voxel response could be like this. It could care less about whether it was intact or maximally scrambled. And of course, it's also possible that a voxel response could be greatest for the most scrambled compared to the most intact, given that, as is obvious to many of you, there is so much edge and high spatial frequency information here compared to the intact. So there may be voxels that are tuned more to those properties than to the integrity of the shape per se. So firstly, this is the inferior surface of the brain, like the eyes are over here, this is occipital cortex back here. And the color, we colored every voxel according to how shape sensitive. So the more blue a voxel, the more shape insensitive it was. Sorry, I hope you can get this. This is like the most scrambled to the least scrambled, and the more red it is, the greater the beta weight to the intact object. Sorry, somehow there are very faint numbers over here that you cannot see. And yellowish greenish would indicate that the voxels don't really care. They respond, but they don't really mind so much what they're responding to. So, as you can see on the ventral surface, we see this slope that's greatest for the most scrambled compared to the least scrambled in early visual cortex, which is exactly where we should see this edge information and the spatial frequency function. And as one moves anteriorly along the ventral surface, we see a region that doesn't really care that much, until we see regions that are in red here, very shape sensitive, both on the inferior surface, and it also folds over to the lateral surface, like to area LOC, for example. So this is what you expect, this is what it should be, and we've got every visually active voxel. We determine whether it's visually active in a separate localizer kind of experiment. But of course the question is, what happens in dorsal cortex? So let me just be sure that this is a very odd view of the brain. So this is occipital cortex in the left and the right hemisphere over here. This is going down the ventral stream, and this is going up into dorsal cortex, the posterior intraparietal sulcus, and up here to the anterior intraparietal sulcus. I hope you can make sense of this. It's such a funny, compressed view here. But really I want to be able to show you the ventral activation that I've already laid out, but I also want you to see the activation going up into parietal cortex and how shape sensitive it is. So again, we have early visual cortex liking the 264 scrambled version, but then there is significant shape selectivity for a large swath from posterior intraparietal sulcus all the way close to the anterior intraparietal sulcus, which is much less shape selective in and of itself. So the finding here tells us that signatures of shape and geometry are not solely the purview of ventral cortex. And in fact, you can see these signatures up in dorsal cortex as well. We also, I didn't include the studies, but the analyses. We also did various other things, like we did multidimensional scaling, or we did PCA, various kinds of things on the data, and we show, at least in the MDS, you can see areas in dorsal cortex are kind of clustered together with areas in ventral cortex, i.e. show the same response profile, even though they belong to these two so-called visual streams. So lots of data that support distribution of what and where in both dorsal and ventral. I haven't shown you the distribution of the spatial information into ventral cortex as well, but it does exist. And so, okay, so we see that there's activation in dorsal cortex. Is it meaningful? Is it functionally relevant? Does dorsal cortex know anything about shape? Does it have the information necessary for object recognition? So together with Vlad Eisenberg, who was a postdoc somewhat recently, we decided to have people in the magnet for a few sessions. We got a lot of data from 5 different categories. Each category has got 5 stimuli, and there's no texture or color or anything that is informative here. And what we want, I'll show you. So they came, they just saw this long stream. They were doing an end-back task if they saw a repeat of 2 of them. And we had this event-related design collecting a lot of data over 8 runs. So we trained a classifier, support vector machine, on just 3 of the 5 instances in a single category, and we tested it on the remaining 2, and then obviously, you know, permuted through all of these. There's a 20-fold cross-validation for each of these different categories. And then we aggregated the decoding accuracy, and as you can see here, there is, so chance is 20% here. There are 5 categories, so it's 5-way classification. And you can see here that 3 of these regions left, sorry, let me just tell you. On the x-axis here is left and right, posterior dorsal cortex and anterior dorsal cortex. And here is decoding accuracy from the classifier over this 20-fold validation. And it's only the right posterior intraparietal sulcus that has significant decoding accuracy above chance. These are kind of hovering around chance, but I don't think we can compellingly argue that they have sufficient information, and so I will only point to the right posterior intraparietal sulcus as having enough information to support object recognition. Okay, so at this point we've got both between hemisphere information and within hemisphere information that shows both segregation of function but also distribution of function. And I need to show you one more thing before we conclude, and that is it's also possible that within a hemisphere there's propagation of signals. So I ruled it out in the case of between hemispheres and the corpus callosum, but of course there are very strong white matter tracts that connect ventral cortex to dorsal cortex, like the vertical occipital fasciculus from Jason Yeatman's work as just one example. So is this really just an artifact of propagation, or is there truly independent shape information in parietal cortex and independent of ventral cortex? So this is a graduate student, and together with Lad Eisenberg, we conducted a study. We had participants in the magnet. We showed them objects, and we showed them scrambled versions of objects, and they also did an end-back task. And we analyzed the functional connectivity of dorsal and ventral cortex. So let me explain what I mean. This is the posterior intraparietal sulcus that I've already shown you is the region where there is object geometric information that can support recognition in decoding. And mapped out here is the functional connectivity, which means the correlation in the bold time course between the seed in the two hemispheres and every other possible voxel in the brain. Okay? So you can see the posterior intraparietal sulcus has got massive connectivity and is correlated with all of these other regions. And the same is true if we put the seeds in LOC, the sort of very almost preeminent object shape region in ventral cortex. Again, you can see the functional connectivity to ventral regions and to dorsal regions. We can see that there are large, widespread neural circuits that are activated when objects are being viewed, both in ventral and in dorsal cortex, and I've already told you that that's the case. But the question that I want to ask here is whether when we see this activation in parietal cortex, whether it's simply the cascade from ventral cortex. And so we did many experiments, but I'm going to just show you one. And that is, we took the data and we simply partialed out the contribution of each pathway when we analyzed the other. So, for example, this is the data from the ventral seed, LOC, and we did not include, we set to 0 everything that was coming from dorsal cortex. So the pink that you're seeing here is the connectivity to the ventral seed without any contribution from dorsal cortex. Think about it as, I don't know, holding constant, no, a better way of saying it is, doing a partial correlation by pulling out any influence from the other pathway. And so you can see that there are regions here, as there should be in ventral cortex. Of course, the more interesting thing is what happens in dorsal cortex when you remove the contribution from ventral. And that is this last piece over here. And you can see here there's no contribution from ventral, and dorsal cortex itself has got pretty widespread and robust connectivity, so showing object and shape sensitivity that's correlated with the preeminent shape selective region in dorsal cortex, even when ventral cortex is held out of the equation. Okay, so It's not an artifact of propagation, not between hemispheres and not within hemispheres. And so we are left now with this, with the claims that if you look closely enough and you look hard enough and you do experiments that contain not just one stimulus type, not just faces, not just words, not just what, not just where, but if you begin to look at the system as a whole within individual, you actually see much greater distribution of function, both between hemispheres and within hemispheres, leading to what I think is a reconceptualization of visual cortex as not binary between or within hemispheres, but much more graded and distributed, with coupling to other areas like to Broca's cortex, like to the superior temporal sulcus, and significant malleability, as we see in the children with hemispherectomy, where a single hemisphere comes to support both types of recognition of words and faces. So, just to close the loop on the opening, the data seem to go against a clear segregation, canonical set of discrete processes account, and favor a distributed gradient account. And so nestled in here, in between pinwheels and retinotopy and whole brain gradients, are gradients that we can see that cross between the hemispheres and also are reflected within a single hemisphere. So I will leave you with the claim that it behooves us to think about circuits, to think about distribution of function and joint activation, the joint contribution from many different areas. The brain doesn't have firewalls. Signals propagate over large, relatively large distances. And if we really want to understand brain behavior correspondences, we need to look at the whole brain, and we need to look at more than just a single stimulus type in an experiment. So the work was done largely by people in my lab. These are all graduate students. The yellow rectangle are individuals whose work I showed the most. Michael Granovetter, Tina Liu on the hemispherectomy, also Sophie Robert, Nick Bluff mostly on the computational work, and Eva Dunless on the developmental face versus word studies. I've also had and have amazing postdocs. This is Eres, who did the shape sensitivity with me, and Vlad Eisenberg doing this functional connectivity that I've just shown you, as well as decoding accuracy from dorsal cortex for object-specific information. We've also had a really good undergraduate student who's worked together with us, and my two faculty colleagues, Christina Patterson, who is the chief epileptologist at Children's Hospital, and we work extremely well studying the children with hemispherectomy. Also these days, the intracranial recordings that we're obtaining from these children, which I haven't shown today. And David Clark, with whom I've done both empirical and computational work. I also recognize the funders, National Institutes of Health, National Science Foundation. Don't ask me if we'll ever get another penny from either of them. And just to confess, I guess, that I'm a founder of a company called Precision Neuroscopics. So I will stop there. Thank you again for the opportunity to give this talk. I also wanted to say that Chris, who I can't see right now, somewhere, oh, right at the back, in the back corner, just before the lecture, came and gave me this token. It's actually a book that belonged to Ian Howard. It has his signature in it. And I feel really moved to have received this. It's not just a random token. It actually feels really meaningful and something that I will take away with me. Thank you very much.
Questions?
First of all, outstanding talk. Thank you very much. I had a question about the hemispherectomy individuals. Did you check to see whether single individuals were highly correlated on the words and the faces, or anti-correlated, or whatever?
We did many experiments. One of the problems of trying to give like a broad landscape talk like this is like I never get into all the controls that we did, the correlations. We did studies because these patients are all hemianopic, and so we either showed the stimuli at fixation, or we showed in the intact field, and we eye tracked them, etc., etc. They are highly correlated in their face and word recognition. I think actually a better way of displaying it is I should have put each individual in a single color. Then you could have seen for yourself that those, as you track those individuals, they are really nicely correlated in their accuracy.
Thank you very much. Just one other question. Reading, of course, is only about 5,000 years old for human beings, whereas we are something like 200,000 years old as a species. What does that, do you think, say about some of the earlier studies with the selective word form area?
Yeah, thanks. Thank you for coming to the talk. Nice to see you. I'm very intrigued by the fact that word reading is really only prevalent in the common culture for about the last 150 years. Prior to that, it was mostly among the clergy who had access to books and ability to read. And so reading, the ability to perceive orthography, is so recent that it cannot be pre-specified in the genome, as some people argue these topographical maps are. We haven't had enough time, evolutionarily, for these to actually be sort of hard-coded and pre-specified and innate and sort of concretized. So that compels me to think hard about the fact that this is not pre-specified in the genome. This is not like genetically, architecturally coded from the get-go. The emergence of this topography and the emergence of these distributed circuits and functions, I would argue, is a combination of visual experience and maturation. So there are wonderful studies, even done today, with individuals who are illiterate or have been illiterate, and mapping out their face and, well, their face recognition. And then as they acquire literacy in adulthood, what happens to word recognition? It basically looks like the data that I've shown you. As I hypothesized in children, like they have two hemispheres that are really engaged in face recognition, and as they acquire literacy, even in adulthood, there's a pressure to couple with language areas, and you begin to see now the emergence of the visual word form area in the left hemisphere. This is work that was done by Stan Dehane. There is also the same kind of study that's done by Manuel Pereira, and I always wanted to do this study, but we were told that we couldn't use, there is a population of illiterate individuals who live in Pittsburgh, and I've been working with the Greater Pittsburgh Literacy Council to access this population. They are mostly women and children who have fled from war-torn areas like the Sudan and Ethiopia, and the State Department have settled them as a group in Pittsburgh. These mothers, the fathers were mostly killed, the mothers are illiterate in any alphabet, in any language whatsoever. And so I wanted to also firstly show they could be like really good at face recognition, because both hemispheres are being devoted to face recognition. Anyway, the bottom line is that we were told that we can't do this experiment because these individuals could not understand the science, and so it wasn't ethical. And then we said, we've got translators, and we're going to pay them $50. Like, you can't pay them $50 because it's coercive for individuals like this. So eventually we threw up our hands, and the studies were published, and they did really great work. And I think that's really important to specifically address this question of what happens when literacy is acquired either in childhood or in adulthood, and how the brain is reconfigured in response to that. Sorry, a long answer.
In a similar vein with the hemispherectomies, did you look at the amount of time since their surgery?
I have 164 slides here. Somewhere in the last 100 is the result that you asked me. So we looked at many things. We looked at the time of their first seizure, the time of their surgery, the amount of time between their surgery and the time that we were testing, because the longer the time, you might expect the more plasticity. The standard, sort of the received wisdom, is that the more time you have and the earlier the surgery, the more plasticity. We had 40 individuals. We cannot see any effect of age at surgery or time of testing. So I think the jury's out, but it's not simply explained on the basis of something about their medical history.
Very cool, thanks.
Hi, you mentioned how for individuals who are illiterate, they would have much more of a capacity for face recognition. So I was kind of wondering whether that pressure created by learning how to read and write would also apply for people who have multiple languages, and whether that would make face recognition harder for them.
Yeah, I've thought about this a long time. So it's a great question. And the question is, if you have to acquire multiple written scripts, either for reading or even just for reading, never mind for writing, just for reading. You're getting a lot of experience, the statistical regularities of that input is significant for both scripts. Let's just say they are bilingual. And the writing might be very different in these cases. So the question is, well, how does the visual word form area cope with all of this? There is no, I do not know of a study that looks specifically at sort of bilingual orthographic representations in the left hemisphere and the extent to which it differs compared to, say, unilingual individuals. There's a very nice and reasonably vast literature on bilingual language and what happens to acquisition of the second language, and it depends on when the second language was acquired compared to the first one, but I have not seen the experiment that you're asking for, and I really like the idea. I think it's worth doing.
Hi, great talk. I have a question about what is distributed and graded. So one view would be that the algorithms are distributed. So you're drawing on information from all over in performing an algorithm. Another view would just be that some of the same contents show up in different parts of the brain. So maybe you have a very localized algorithm for identifying shapes that's right in the ventral pathway, and an entirely separate algorithm for for acting on shapes. But it turns out you need to represent shapes both for each of those separate algorithms. And so do you have a view about what it is that's distributed exactly?
Yeah, super question. I think all I've shown you today is that that content is shared across multiple areas. I think it's not that I'm not convinced that this is a product of different algorithms, and that's probably because, and I didn't show it to you, that in this study, we did all of these comparisons about the exact nature of the neural signal in dorsal versus ventral cortex as a function of the scrambling of the stimuli. And as I mentioned, doing things like clustering algorithms and all the acrobatics of advanced analytics that you can throw at this, they actually look really similar to each other. So maybe there still are different algorithms, but I think to be conservative, I'll say that they are representationally highly similar to each other. So, David Plout and I right now co-advise a neural computation student who is essentially doing modeling to understand the question about algorithmic differences in the two pathways and the extent to which different architectures in a sort of artificial neural network give rise to different kinds of findings in these two pathways. So I'm hoping I might be able to tell you something more from looking at these convolutional models about whether the algorithms themselves are different in the two pathways. We do know some things, like the receptor field size in ventral and dorsal cortex are actually different. And so you might think that that's got to play a role in all of this. It's not like you would always get something exactly the same, where the physiology actually differs, the antecedent physiology differs. So I think the jury's out. I wouldn't be surprised if there were some algorithmic differences between them. Having the same algorithm, like just replicated, seems like a bit of a waste of time and money in the brain. So maybe I'll have a better answer for you in time, but I'm being conservative at the moment, really only talking about the representational similarity.
Thanks.
Yeah, thanks, Marlene, for a wonderful talk. My impression, my question for you is where do we search next? Because we are searching for something. Because, like, it seems every time we are looking for something specific, like word processing or face processing, we figure lateralization is very limited or absent at all.
Yeah.
But on the other hand, there is very, very strong and reliable asymmetries.
Yeah.
For instance, like one that's old and has been described a lot is hemisphere, like semi-neglect, for instance.
Yeah, sure.
If you, like the right hemisphere seems to be much more general.
Correct.
If we lose the right hemisphere, like bad things happen. If we lose the left hemisphere, the right hemisphere apparently can make up for it. So that's why we hardly ever find like right-sided hemi-neglects or after left-sided lesions. Or whatever the lateralized, like more general functions like animate and social on the one hand versus inanimate and piecewise, which go much further back, or which are found not just in humans but in other primates as well, but also in birds, for instance, like pigeons and chicks use their right eye more for their right eye more for their left eye more for social encounters and stuff like that. So there's motivation to search or to try to investigate what drives those large high-level differences. Where would you look next?
Well, first, let me just be sure that that my message was clear. I'm not saying there are not asymmetries. There are totally asymmetries. There are asymmetries for faces and word representation, for language and spatial functions, for social functions. There are asymmetries, but they are not absolute. We're not talking about binarization of function here. We're talking about graded representations. In the summer, I am going to, I've become really interested in understanding more about these hemispherectomized individuals. What happens, what are the capabilities of a single hemisphere? To what extent and how can it accommodate functions that would have been served by the other hemisphere? And does it matter if it's the left or the right hemisphere that's preserved? So in the summer, I'm going to be examining whether or not these children have language deficits versus spatial deficits, depending on which hemisphere they have preserved. What I showed is that a single hemisphere can get up to 85% function for faces and word discrimination, and it didn't matter which hemisphere it was. So the question I asked myself is, well, is this true for other sort of seemingly asymmetric functions like spatial functions, attention functions. No question, hemispatial neglect after a right parietal lesion is so much more common compared to left parietal lesion. So we know that there are differences, but is this plasticity, is this ability to have what seem like binary functions appear sort of interleaved in a single hemisphere, but not completely as accurate as you see in individuals who have got two hemispheres. There has to be a cost if you've got a single hemisphere. Two hemispheres are better than one, no question. But I'm hoping to be able to understand whether or not this sort of plasticity, this malleability in the single hemisphere extends beyond just occipitotemporal functions in service of face and word recognition. So I can let you know. The interesting thing is that in the summers, every second summer, there is a large organization of families of children who've had these major resections. And it's very complicated medically. And so there are 100 families who come to these summer meetings, and the organization, which is called the Pediatric Epilepsy Surgical Alliance, is extremely kind and allows me to test patients at the conference, which is why I can get 40 of these participants tested in a single weekend. So I'm hoping to get another 40 when I look at spatial and language functions, and hope that in the not too distant future I'll be able to say something more meaningful rather than speculative about this.
Thank you. Terrific talk, I'm convinced.
Oh, really? You shouldn't be.
But I have a question.
Of course.
I'm curious, to what extent does the organizing structure, let's say in the left hemisphere, the region and around Broca's area, to what extent is it necessary to continue operating to keep, let's say, the word form area in the posterior part of the temporal stream isolated, not isolated, but localized in some sense? So in other words, a patient with Broca, who has a Broca's aphasia or an anterior aphasia,
Yeah.
would that patient also show a word form, sensitivity of a word form area, or is that lost? Is it there only, in other words, to organize it and afterwards it doesn't need it?
Yeah, I understand the question. Before I answer that, let me give you a little bit of evidence that I have, and that is that in these hemispherectomy cases, Broca's, in an individual who only has a right hemisphere, the left hemispherectomy, Broca's area emerges in their right hemisphere, and the visual word form area emerges in the right hemisphere. So there's definitely something about the co-localization of these two functions that is important or is compelled if you've only got a single hemisphere. So in somebody who has Broca's aphasia, is the visual word form area, so here is a possibility. The visual word, Broca's area is essential for the initial structuring of the visual word form area. It's that wiring and that coupling that sets up the left hemisphere for literacy. But Broca's area might potentially then go away, and you might still be able to see activation in the visual word form area. Not so much meaning, not the linguistic aspects might go away, but certainly the orthographic representation, the perception of the input.
That's what I'm asking.
So I don't have the data specifically, but we know, for example, in adults with deep dyslexia, who usually have a lesion to Broca's area, they can still read. They just they read the word peach as apricot. So they certainly have represented the input, but the language aspect, the semantics, has sort of gone awry. So it is my contention that Broca's area is necessary for the initial implementation of the visual word form area. I'm conjecturing that Broca's area could then go away, and you would still be left with isolated but functional visual word form area in the left hemisphere. I don't know. Great experiment. We should find some patients.
Go all the way to the back, so it'll be a second.
Thank you very much for the great talk. So my question is about if we can find more elementary processing which can explain the result. And that could be whether there are differences in the pattern of eye movements. Because, for instance, we know that in individuals with autism, if we look at their brain responses to faces, it's totally different. We may not find fusiform face area, but when we normalize for the eye movement pattern and we force them to look at the same places that other individuals look at, then we get the same pattern. So here for words, we make eye movements from left to right. For faces, we make different pattern of eye movements. We can even test different languages like Mandarin when we make eye movements from up to down. Is that something which could explain that there is no high-level categorization in the brain, or it could be something more elementary or not? Thank you.
Great question. So yes, of course, you know, the signature of eye movements in individuals with autism is really quite different, especially in response to faces, compared to, say, geometric forms, compared to non-autistic individuals. So we have two other lines of work, one of which is studying the eye movements of individuals with hemispherectomy, for example. We know that they have differences in eye movements. It has to do with the fact that they are hemianopic. It's not stimulus type dependent. It's not the eyes or the mouth. This is just a blanket difficulty in both saccading and also in smooth pursuit to the side opposite the lesion. We also know that if you hold eye movements constant, like present stimuli solely at fixation, and you have an eye tracker on them, and if they break more than a degree or 2 degrees of fixation, you abort the trial and do it again. The results still hold, even when everything is held constant at fixation and there is no eye movement requirement at all. So I don't think what we're seeing here is a sort of functional consequence of an eye movement profile or an eye movement difference between the stimulus types. I think it really is something to do with the stimuli themselves.
Hi, Dr. Behrmann. Thank you so much for an incredible talk that really spans, many years of incredible research that you've been a part of. And you mentioned at the end being involved with a company. You kind of disclosed it a little bit. And I was curious if you're comfortable to talk about the way that your research has led you to potentially working with industry. I know that research nowadays is really moving a lot more towards applications, and just given the nature of your work being so fundamental to our understanding of the brain, if I know there's a lot of trainees in here today who might benefit from a little bit more of how the work that you've done has led you into paths outside of academia.
Yeah, thanks. It's really a great question. This topic came up at lunchtime as well when I was talking to some of the trainees, and I think it's particularly pertinent in a place like CVR, which has a strong translational component as well. So the way that this company was founded, Precision Neuroscopics, is when I was at Carnegie Mellon University. I recently moved to the ophthalmology department in the medical school, but when I was at CNU, there's like every second person is an engineer, and some of my best friends were engineers, and so we started running experiments together, and in particular we started thinking about ways to improve both the hardware for better acquisition of brain data, and also the software and the analytics that could be used to interpret the brain data. So the company actually has got two arms, a hardware and a software arm. The hardware sort of came about because when we started thinking about this, we wanted high resolution EEG. There are many places in the world that don't actually have MRI scanners. So could we use high-resolution EEG, which costs a fraction of what a magnet costs, and sort of deploy those in the field and have algorithms that, for example, can detect lesions in the brain based on scalp EEG. So we did two things. The one is we developed new kinds of EEG caps that are high resolution. So of course, people usually have 128 electrodes all over the scalp. We decided we were going to put like 100 of those electrodes just around visual cortex and sort of going to temporal cortex. The first time we ever did it, like there were so many electrodes, the cap was just like a straight line at the back here, so that obviously didn't work. We eventually 3D printed the holders that you had put the electrode in, and so we could actually get the holders in, we can get the electrodes into the holders, and then it was sort of conformed to the shape of the head. We showed that using that kind of acquisition, very high resolution, super Nyquist acquisition, so minimal space between the electrodes, we had much more precision in decoding the visual stimulus than a standard EEG cap, almost of any configuration. So that was one hardware development. The next hardware development we did was, it became clear to us that trying to collect EEG data from individuals whose hair is quite coarse, so for example, African American individuals, you can't actually get good contact with the scalp because the hair is much more springy. So it's like, oh goodness, like nobody is collecting good EEG data. There's like a whole massive population that isn't as good healthcare access because there isn't decent EEG capabilities. So we developed a different kind of electrode that can be used with people who have dreads or with braids, called Sevo. The company is manufacturing those. There's a lot of interest in having those kinds of electrodes, both for science but also for medical applications. Of course, EEG is like a standard thing in the hospitals for individuals with seizures. And on top of all of that, we developed algorithms, including something that's called silence localization, in which we can show that using 3 minutes of scalp EEG in one of these fancy caps, we can detect regions of neural silence, which we also call a lesion, and you don't need to use a structural MRI to be able to do so. So that algorithm is also being sort of distributed as part of the company. So I never imagined myself ever working in a company or having anything to do with a company. It was always like those other people who have companies. It's turned out to be a really thrilling experience. I actually love having had this exposure to markets and tech and hardware and software. It sort of opened my eyes in different ways, and I'm very grateful for that. There's a tremendous need. We sort of are at a really interesting juncture now, I think, in tech for medical usage in many other ways as well. And I think the kind of skills that one learns in something like cognitive neuroscience can be well utilized in some of these adaptive and new technologies. Thanks for the question.
Fantastic. So I think we could probably keep you talking all afternoon, but maybe we should give you a break and have a discussion over a glass of wine or a cup of coffee. There will be some snacks brought in a few minutes and some drinks, so please stick around, and please let's thank our speaker. Thanks everyone.